This is the microbiology laboratory. This is specimen reception. Here all the samples come from various hospitals within the trust and other users of the service. Samples are sorted out by sample type for the various benches in the laboratory. We have the urine bench, the feces bench, the wounds and fluids bench, we have a gynae bench, we have respiratory sections. So the samples are coming to the reception, are checked and opened up and checked that they correspond to that particular patient, and then taken through to the various sections in the lab for processing. This is our rapid MRSA screening bench where patients' nose swabs are screened to see if they carry MRSA. The detection method used is a PCR method, which is a polymerase chain reaction method, looking for the genetic material of MRSA. This laboratory does the testing for the sexually transmitted infections chlamydia and gonorrhea. Chlamydia is the most common sexually transmitted infection. This is the Tigris GenProbe analyzer, which carries out the molecular detection of the nucleic acids of chlamydia and gonorrhea using a process called transcription-mediated amplification. This bench is the wound and fluid culture bench. Here we get samples such as wound swabs, abscess swabs, leg ulcer swabs, bite swabs, tissue swabs, fluids from the chest, pus samples. The samples would come through to here. We would check the demographics and the details of the sample, and then culture the sample onto various media using sometimes basic nutrient media such as blood agar and other selective media dependent on what we want to grow from that particular sample. We also do microscopy on those samples, on specific samples. We would do gram stains and look for the bacteria under the microscope. This is the CSF bench where we receive cerebrospinal fluids to help in the diagnosis of meningitis. CSF would come and the biomedical scientists would need to examine the CSF directly and then do a cell count on the CSF using a counting chamber. The CSF is also cultured and microscopy is performed. This is the feces bench. This is where we process stool samples for the causes of food poisoning, such as Salmonella, Shigella, Campylobacter, E. coli 0157. We also look for Vibrio cholerae. We would also look for causes of antibiotic-associated diarrhea. We would test for Clostridium difficile. We use various culture media, very selective agar, to look for Campylobacter and Salmonella. And then we would follow up culture growths using various biochemical tests and serology tests. This is the blood culture room. Patients' blood culture samples are taken when they suspect bacteremia or septicemia. The blood culture bottles are loaded onto a blood culture analyzer, and positive samples are flagged, and then microscopy is performed on those samples, and those samples are cultured, and then antibiotics are tested against the organisms that grow. This is the day one wound bench. This is where all the cultures that were set up on the wound and fluid bench are read. Plates are examined and the colonies identified using a variety of commercial identification tests, such as the Staphyrex test, and biochemical tests such as the bile-esculin test and DNase test. These plates show manual susceptibility tests by the BSAT method. This is the genitourinary medicine bench. Here we would culture vaginal swabs, cervical swabs, urethral swabs for the causes of vaginal discharge and urethral discharge and sexually transmitted infections such as gonorrhea. This plate shows Candida albicans, which is a cause of thrush. This plate shows Neisseria gonorrhea. We would also do microscopy for a parasitic infection called Trichomonas vaginalis and an infection called bacterial vaginosis. We also have a neonatal screening bench where we screen newborns for the signs of infection because they're immunosuppressed, so they need to be regularly monitored, and if they've got signs of sepsis, then screening swabs and other appropriate samples will be sent. This bench is our antibiotic susceptibility testing bench where we would do manual BSAC susceptibility tests using paper discs. We would also do E-tests. This test is a fluconazole E-test for Candida species. We would also, on this bench, monitor serum levels for antibiotics such as gentamicin and vancomycin to check that the level in the serum is not too toxic and that it is at the right therapeutic level. We would also do antibiotic susceptibility tests on the organisms that are significant, And some plates we would have to re-incubate for additional incubation, and some plates do not come out until after 48 hours incubation, such as anaerobic bacteria, and this plate shows an anaerobe which is sensitive to metronidazole. Another way of doing susceptibility tests is by using our Vitek 2 analyzer, which is an automated identification and susceptibility testing machine. It uses identification cards to help identify the organism, so it would tell you whether it was an E. coli or a Staphylococcus aureus. And it uses susceptibility cards which have antibiotic in them, and the bacteria will grow in the antibiotic or be inhibited by the antibiotic, and the machine interprets both sets of results by comparing the results to its database. This is the urine bench. This is where we use to diagnose whether a patient's got a urinary tract infection. We can do microscopy directly on a urine using an inverting microscope to look for white cells and red cells and bacteria. We also culture the urine sample onto chromogenic agar, which changes colour dependent on the organism that grows. And this plate shows an E. coli, which is the most common cause of urinary tract infections. We also do screening on this bench for MRSA by culture, but our mainstay screening method for MRSA is PCR. But we can culture swabs for MRSA on special selective media called Brilliance agar, and this plate shows denim blue colonies of MRSA on that agar. This bench is our respiratory bench. Here we process samples from the upper respiratory tract, including ear swabs, eye swabs and throat swabs, and samples from the lower respiratory tract are processed in containment level 3, and samples include the sputum samples and bronchoalveolar lavage samples. They are processed at containment level 3 because there is a risk of the sample containing tuberculosis. Throat swabs are processed for organisms such as group A streptococci, ear swab Haemophilus infections, and eye swabs you can have Staph aureus conjunctivitis. This plate shows a group A streptococcus from a throat swab. After culturing, we need to incubate the plates to grow the bacteria. Some bacteria like to grow in oxygen and at a temperature of 37. Other bacteria like to grow in a carbon dioxide atmosphere, and we have incubators that have CO2 that consistently flush through the incubator. And specific bacteria are what we call anaerobic, and they grow without oxygen, and therefore we use an anaerobic incubator which has the oxygen replaced with a hydrogen and nitrogen gas mixture. We also have a walk-in cold room where we keep all our culture media, all our antibiotic discs and all our reagents so that they're refrigerated. We also keep the samples for between a week and a month in case we need to go back to them for reprocessing.